Hi, welcome to the bioinformatics.chat. My guest today is Michael Love. Mike is an assistant professor at the University of North Carolina, Chapel Hill, and there is a good chance you know him as the author of DESeq2, which is one of the more popular packages for differential gene expression analysis. Mike, welcome to the podcast.
Thanks so much. I really enjoy listening to these, so it's a pleasure to be here today.
Awesome. And let's maybe start with your background. So how did you start your career in bioinformatics?
Yeah, so I was I think when I first became interested in genetics and bioinformatics, I was doing a master's in statistics at Stanford, and some of the courses touched on computational biology and bioinformatics. And I did an internship over the summer at a microarray lab at UCSF. So it was the lab of Prescott Woodruff, and they were looking at RNA expression in patients with asthma and smokers and then non-smokers. And that was incredibly interesting to me that you could see differences across patients at the gene level. So then from there, I thought, well, this is where I want to apply statistics, and so I looked into computational biology PhD programs.
Mm-hmm. And which one did you find?
It's kind of funny. Like, it was very directly from when I was in this internship, and also other times during the this master's program, I had used a lot of bioconductor packages. So I was already kind of in this space of thinking, well, I like that there are people who are statisticians who are developing software and then making it broadly available. So I was kind of already clued into bioconductor as a way for methods to be pushed out. And then so I think the way I found the lab that I was interested in was I looked up one of the packages that I'd used in that summer. And so it was the, among other packages, there was like this variance stabilization package that came from Wolfgang Huber was first author, and Martin Vingren was the last author. So like I zeroed in on the Vingren lab and saw that they had a computational biology PhD program, which was like supported by the Max Planck Institutes. And so that ended up being like one of the ones that I was most interested in. And then I applied there and interviewed and got in.
And you mentioned bioconductor, and I don't know if everyone is aware, everyone is familiar with what that is. So maybe let's explain that.
Sure. So it's a number of things. I'm teaching computational biology right now at UNC. So I think a couple of weeks ago I had this talk. It's a lot of things. Many people might know it as a repository for R packages. So it's an alternative place where you can download R packages. But it's a project that goes back, I guess, like more than 16 years now. That was, started by a bunch of statisticians, academic. I think it's primarily academic statisticians who are interested in writing code that could work together. So, like, each person works on their own method, but if we agree upon some like data structures, then we can more easily have our packages work together. And I think it was predominantly like microarray was the driving force behind wanting the packages to integrate. So it's a group of, it's a project, it's a group of people that work together. And then it's also a funded project. So there are U grants from the NIH and elsewhere now. It's broadened its funding that helps support the project and allow there to be not just the individual developers writing packages, but there's also a core team that kind of supports the core set of packages that provide algorithms and infrastructure that's kind of like foundational.
Were you already predisposed towards R from your statistics program?
Yeah, so let me think. There was, in statistics, it was pretty much all R. There was like, like among the professors there, like, people like Hasty and Tipsharani and Efron and writing a lot of R packages. So there's always a lot of open source development in R. And then I had taken a class taught by Naris and John Chambers. Like my statistical computing class was taught by one of the person who invented S, like the predecessor to R. So it was pretty heavily like slanted towards R. I don't think there was any Python in my master's program.
Have you ever regretted throughout your career? Have you ever considered like jumping across the fence?
I've been I think Python is well. I now that I'm teaching, I mean R is not very easy to. There are definitely like because it's created by lots and lots of different people, and there's not like maybe less of a top down design structure. Like argument names are very different across packages because people have different preferences for that. So there is aspects that makes it more difficult for teaching, where I think I could imagine it being a lot easier to get people started with Python. I think also that one place where it's clear that there's a lot of appeal to Python is with the single cell community. So a lot of the like key single cell processing and analysis packages are Python, are coming from Python. So
maybe because of the machine learning influence.
Yeah. Oh, and of course, and also for like deep learning. So at UNC, the biostat students that get into deep learning will often a substantial part of their research is done in Python.
So you went to do a PhD at Max Planck, and was that where you worked on DESeq2?
So I did. I worked in the Wingren lab predominantly. I also had a spot in Knut Reinert's group at Freie Universität in the informatics. And for, I guess, like the first 2.5 years of my PhD, I was mostly working with sequencing data, and I had noticed the Huber group in Heidelberg at EMBL. And so I was really lucky, I think, that I was in a position where I asked Martin, my primary advisor at that time, if I could like go study for some months, like as a visitor to the Huber group in EMBL in Heidelberg. So that was like, I was down in Heidelberg just as a visitor. And then that was like one of the chapters in my dissertation. So I started my PhD in 2010. And the idea of sequencing the cDNA. So like we're interested in measuring the RNA in samples, and previously, like what I had done in my master's program was look at microarray measurements, which are these continuous values, basically like the intensity of a fluorescence from probes on an array. Then in between that time, between my master's when I started my PhD, the papers had come on around 2008, which showed that you can, you convert the RNA to cDNA and then sequence the cDNA in these DNA sequencing machines, which are at that time, getting better and better. The throughput and read length are both getting better and better. The error rates are going down.
Where cDNA stands for complementary DNA, right? So maybe explain what.
Oh yeah, so there's just a reverse transcriptase to take a single-stranded RNA to get that to a double-stranded, what's called cDNA, because it's complementary to you're forming the complement to the single-strand RNA you get from the sample.
Mm-hmm. And the idea is that at least back then we weren't able to sequence RNA directly, right? So we would reverse transcribe it to DNA in order to sequence it.
Yeah, it's basically like, at that point, I think in that time range, the power of the DNA sequencing machines and their rapid improvement in throughput and read length was revolutionizing like functional genomics, because we can sequence RNA that is converted to DNA. We can sequence DNA that is attached to proteins with immunoprecipitation. So just using that machine to make measurements was changing all of the fields. And I was in the Max Planck Institute in Berlin, the molecular genetics Max Planck, they had also been one of the earliest RNA sequencing sites. So there's a paper from Marcel Schultz and a number of other groups, and including the Vingren department. They had been one of these early RNA sequencing papers in 2008.
Right. And so we reverse transcribe RNA to cDNA and we sequence it. And what's the whole point of this exercise?
So we can then try to assign those sequenced reads to gene sequence, and we could map them to exons or to the genome, and then so allow for spliced alignment to the genome, and then obtain a count. So we're trying to estimate the number of observations of each gene, and so one thing at that early step is the ambiguity. There could be ambiguity in assignment, but if we put that aside for a moment, obtain a count of reads that can be assigned to genes, you can convert this. You can think about this as like a table. If you have multiple samples as columns and multiple genes as rows, you have a very tall table of data. And basically interested in comparing the abundance of genes or exons or isoforms, whatever your features of interest are, you're interested in comparing those across samples.
Okay, and that seems easy enough. You just compute the average, right, across samples or across groups, and just compare, or maybe use a t-test. What's the problem with that?
Yeah, exactly. And I think there were a number of papers that just went ahead and maybe log transform the data and then apply t-tests. So the issue is at the time, and still there's an interest in having as few replicates as possible. So like it would be very common to have two biological replicates in early 2010s per condition, if that. Sometimes there are experiments with no biological replicates, and so if we set aside the no replicate cases, even if you have two replicates, you may have some chance at getting like the sign of the effect size right, but the variability within replicates, among biological replicates, you have very little sense of what that within group variability is. So you have an estimate, standard deviation, like you could pool the estimates of standard deviation across the two groups. That's like the pooled t-test, but it's very noisy. So you shouldn't really trust that estimate. And so conceptually, like what should we do? Like what should a statistical procedure do if it's very uncertain? The t-test does take that into account, right? It widens the distribution. So the null distribution for the t-statistic takes into account that we have few degrees of freedom. But there had already been this, I guess it's a good point of the conversation to bring up. There had already been statistical methods for microarray data that dealt with few samples. So like limma was a very popular method from Gordon Smythe's group, and that had already come up with a framework for what to do when you have thousands of features like genes and very few replicates within which to estimate the variability. You can form a prior distribution on the variance, this within group variance, by looking across lots and lots of features. And then you can apply a Bayesian technique to update the information about the variability for each gene.
So there was limma, and correct me if I'm wrong, but I think some people even sort of tried to apply limma to RNA-Seq data as it was.
Yeah, there may be some in that range. I guess at around I know there's a paper there. So there's a Mark Robinson and Gordon Smythe had a paper for introducing EdgeR. I think I'm thinking like 2009, and then maybe even some other papers about estimating variability when you have counts instead of continuous valued data. So like, comparing different estimates of dispersion. So we use a different term when we talk about count data than continuous. Sometimes we talk about dispersion. So I think, there was already a sense that we should take into account the different data distribution that we're getting with sequencing data as compared to with microarray.
Abstracting from the distributions for a minute, if we have some kind of distribution, some kind of statistical model that describes our data, whether it be microarray data or RNA-Seq data, what are the different options to do the analysis? So you mentioned a sort of Bayesian approach where you have a prior distribution on the variance. And maybe let's even explain, like, what do you do with the variance? So let's say you're able to estimate the variance. How does that fit into the whole analysis? How does that help you?
Yeah, it's a great question. So there are varying approaches even then. The t-test takes into account, it widens the null distribution based on the understanding that the estimate of the variance is not a fixed quantity. So you estimate the variance. You don't obtain it like from an oracle. I like to talk about oracle with students because it's like, well, yeah, imagine we just got the true value of the variability for each gene from an oracle that told us this is the true value. Then the test statistic would look like what? So the t distribution already widens when you have few samples to take that into account. Another thing you could do is just assume that we know it, and then you would have a z statistic or a z-score. So, using the t statistic is already a little bit taking into account the fact that you have fewer samples. There are varying frameworks in RNA-Seq to whether or not to take into account the fact that we estimate the variability.
But once we have the variance, whether estimated or assumed to be from the oracle, what do we do with it?
Yeah, so then you can plug that into a distribution, and you can calculate a likelihood. So we can then calculate the likelihood of the data given our estimated parameters, and then we can form tests. So we can say what we can estimate our parameters if there is a difference between the say groups of different samples. So let's say just two groups for simplicity. So we can estimate parameters, a location parameter like the average that you mentioned, and then the variability. What would those parameters look like if we think that there's a difference? And then we can also estimate parameters supposing that there's no difference. So that's called the null hypothesis. And then a very good approach and popular in statistics is to form the ratio of those two. So likelihood, just like plugging in the parameter values and calculating the probability for a certain distribution, and then calculating the ratio of those two. We automatically there is then a procedure for evaluating the likelihood ratio statistic. So we have from statistical theory what the distribution of that would be for various scenarios. And so the likelihood ratio test is a very popular test at that point.
So do all of these packages, so DESeq2, EdgeR, limma, do they all do more or less the likelihood ratio test?
Yeah, I think I would say it's definitely implemented in all the packages for various reasons. When we were writing the DESeq2, the default test that we perform is a different test. It's the Wald test, which is the ratio of a coefficient in our model, a parameter in our model, divided by its standard error. So we actually do a different test by default, and we had some reasons for doing that are maybe technical, but we also offer the likelihood ratio test, and it's encouraged for certain particular designs. But I would say another important framework, which is not in DESeq2 but is in this EdgeR package, is the idea of a quasi-likelihood. And so they have some additional theory there behind what's called a quasi-likelihood. And I think the key thing for people to know about quasi-likelihood is that it takes into account that the variance is estimated and not known, which is a very, it's a very good thing to take into account, especially when you have small degrees of freedom.
So I think the point that we've made so far is that it's important to be able to calculate the likelihood, and for this you need to be able to describe your data. You need to write down your statistical model, and those models differ for microarray and RNA-Seq data. So, and the reason we should clarify the reason they're different is that it's bad form to describe count data with a continuous distribution such as normal. And so probably one of the first distributions you would consider for the task of describing discrete data is Poisson, right?
Yep.
What is the issue with the Poisson distribution?
So Poisson is a really good distribution for comparing technical replicates of sequencing data. So one little exercise I have in my when I do tutorials or teaching is that you can download technical replicates where you continue to sequence the same library, and that's really well approximated by a Poisson. I can kind of walk through how that happens. Actually, like why does it end up looking Poisson? So you have a fixed set of molecules when you prepare a library, and your sequencing is then like sampling from them with you have a very large number of samples, and each the molecules could be, you can imagine them colored by the gene of origin. So you're sampling from this big pool of molecules, and that's essentially a multinomial sampling process. So there's sampling variability, and if you think about each gene, so like each color in this sampling process, that's going to be a binomial. If you just consider like what is the count for this one gene among thousands, it would be a binomial. And the Poisson is a really good approximation for the binomial when the probabilities are small. So like one gene relative to all the others is pretty small, and when the number of observations is big. And so we're in that framework, we're in that realm when we're doing RNA sequencing. So that's why you get this Poisson variability. If you look at technical replicates, then the issue is, why not just use Poisson? Is that when we look at different samples, say from different mice, these are all like in the same condition group, like all control mice or all the treated mice. We're no longer sampling from a fixed set of molecules, but the molecules, the proportion of each molecule is not fixed across the mice, across the biological replicates. So the fact that P, like the probability vector from which we're sampling these molecules, is changing, that induces over dispersion. And that just means more variance than something, more variance with respect to something. And in this case, we're talking about the Poisson. So more variance with respect to the Poisson.
Yeah, so the thing to explain here is that unlike, for example, a Gaussian distribution that has a separate parameter for the mean and separate parameter for the variance, the Poisson distribution has only one parameter, and that parameter determines both the mean and the variance that I think are equal. So you can independently specify your mean and the variance.
Exactly. Yeah. So the Poisson has this one parameter, and it comes from, it's the limiting case of the binomial. So before we're talking about like sample in molecules and they each have a probability. So the limiting distribution is lambda. The single parameter of the Poisson is P times N. So you would expect, if you sample 1 million fragments and you have something that's 1 over 1,000 probability, you would expect 1,000 count would be your average, and you can plug it into the Poisson, and that the standard deviation would then be like the square root of that. So the square root of 1,000 is how much like you could expect lower by square root of 1,000 or higher by square root of 1,000 around the mean value of 1,000. If you change P, which happens when you look across mice, then you're changing the lambda. And so then you will get more variance than just the square root of the mean value.
If the Poisson distribution doesn't work there because it's a single parameter distribution, it doesn't have enough degrees of freedom to account for the varying lambda between the different mice, let's say, what do we use instead?
So you would start to think of a mixture distribution, because you have a sampling process, and if P was fixed, once you fix P, once you fix a set of molecules, then we can see that we have a sampling process that's well approximated by a Poisson. The mixture part is you have to account for the variability on P. And so you could actually formulate multiple distributions for how P might change across samples, or P times N, the lambda, how lambda might change across samples. So what are our restrictions? It has to be positive because it's an abundance. So you can't have negative abundance. But that's about it. Like, I guess as long as it's continuous valued and non-negative, then any distribution with support, meaning that it allows values in that range, is allowed.
Right. Well, one thing we should clarify is that often when people talk about mixtures, they mean finite mixtures. So you have lambda 1, lambda 2, lambda 3, but that's not what you propose here. You're saying let's take an infinite, let's mix an infinite set of distributions.
Oh, yeah. I guess maybe I should say a hierarchical distribution would be another way to say it. And it basically means you draw one parameter from a distribution, and then you use that, and then you draw data from that draw. So there's kind of, you're going to use two distributions to create data, let's say Y, like our data will be Y, and we'll first draw one parameter, and then we'll plug that in to another distribution, and then draw data. So yeah, sometimes it has multiple names. That could be called a mixture distribution, or maybe it's better to say hierarchical distribution, because there's a hierarchy of these two things. And if we put Poisson at the bottom of our hierarchy, like that'll be the last thing we do, is we'll get data that's count distributed, and it will have sampling variability. You can put something above that tells you about the variability of P, the proportion of each gene across the mice. And you could put multiple things there. So like one thing you could do is log normal. So like it could be that is continuous valued and allows different scale and different shape. Or another one that ends up being useful is a gamma distribution. So it's another positive valued distribution that allows flexibility in how much P changes across the biological replicates. And I guess the important thing for the math is that it's conjugate to the Poisson. That and the simplest explanation of what I mean by conjugate is that we can write out a probability distribution for those two distributions combined. So if we pull a value from a gamma distribution and we plug that in as the rate of a Poisson distribution, that resulting distribution, if we tie these two things together, we can write out in a closed form the mathematical formula for the probability of a given value, of a given count at the end. So that's then a really useful thing, because we can calculate likelihoods in a closed form setting.
Yeah, and that's called the negative binomial distribution.
Yeah, that's called that. So Wolfgang Huber has over time tried to start calling it gamma Poisson, just because it's so much better for teaching. But that's what, gamma Poisson and negative binomial are equivalent. And we use negative binomial in the DESeq2 paper in software, but those two are equivalent.
Yeah, and I think over time, negative binomial, I don't know who was the first person to propose it, but over time, I think it became very popular for these types of data. And are you actually aware of anyone using anything except negative binomial these days?
You might often see Poisson. So there are Bayesian models, Bayesian hierarchical models, where they basically just construct a similar setup to the gamma Poisson. So you might I just saw we just had Christina Kondrarsky from University of Wisconsin give a seminar last week at UNC. So like in my mind, I'm thinking about she was showing a single cell RNA sequencing model, and she had Poisson for the counts that we observe. But then there's more things going on that explain how the rate varies across cells and across cell types. And she mentioned like this gives you something similar to what you would expect with negative binomial. So there'll be variability. It's not fixed within a cell within a cell type. There's extra variability going on, so you'll get this over dispersion. But she didn't write out this is we assume the data follows a negative binomial.
Right, right. And we'll talk about Bayesian approaches a bit later. But I guess what I meant to ask was that it seems this conjugate behavior with between the gamma distribution and the Poisson is so much convenient that no one really considers like using log normal plus Poisson, right? Or
they do in I've seen it in microbiome research. So there are cases where of count sequencing data or count data in high throughput count data in genomics or metagenomics where this gamma assumption of the variability of the probability vector is not sufficient, and so that there's somehow it doesn't give you a compatible distribution with the data. So I have seen log normal Poisson in microbiome.
very interesting. Okay. And so then the question becomes, how do you estimate the parameters of that higher level distribution of that, for example, gamma distribution or log normal distribution, right? How do you do that?
Yeah, so we can, the first thought is to maximize the likelihood of the data. So in statistics, that's called the MLE. If we try to estimate the value of how variable things are across biological replicates, by multiply. We have this closed form likelihood. We can multiply it across the samples and just maximize it. That gives us the MLE, and with lots of samples, that will be a good estimate. That will be a good estimate. So it'll get close to the true value if we think that the data follows that distribution. But we're not in that realm yet. So we have too few samples that the MLE is close to the true value.
So that's even taking into account that you have lots of genes, or just having lots of genes not help there?
Well, so it doesn't help yet. So when I say like we can maximize the likelihood, I should be more specific. I should say if we look at one gene at a time, and we try to estimate the variability within replicates. And even let's make a really simplifying assumption that the variability will have the same distribution in the two groups, and that the only thing that changes is the location. So the only thing that changes is the expected value, but that we'll have some assumption that the way that the variance is linked to the mean value will assume is the same across the two groups. So even if we make that assumption, we still don't have enough information to reliably estimate that these parameters for the gamma with like 3 samples per group.
And that notion is a very important one, the mean variance relationship, right? So we already discussed it in the context of the Poisson distribution, but maybe you can describe it more generally.
Sure. So when we specify this hierarchy of distributions, like the gamma for the rate of the sampling process, and then the Poisson to get the count data, that induces a relationship between the variance of the data and the mean value. So they're linked by a parameter which is called the dispersion, and it's actually parameterized in multiple ways, but the one we used where if the dispersion is higher, the variance is higher. So it links it in a positive way. And as the counts get high, it's basically that the variance is a multiple of the mean squared.
Mm-hmm. But that's specifically for the gamma Poisson distribution. That doesn't hold for other kinds of priors.
Right, right, right. So that's specifically once we assume this relationship, then we have this link between the variance and the mean, and if we're specifying that each gene has its own dispersion parameter, then each gene can have kind of has its own link. So we get the dispersion estimate for this gene, we get the expected value for these samples, and then we can plug in what we think the variance will be of the counts. But we could allow that dispersion parameter to vary across the genes. Sorry.
Right. And so what you were saying, I believe, is that if we assume a unique dispersion parameter for each gene, then we don't have enough data to estimate that dispersion parameter and plug it into our likelihood function and construct the likelihood test, right? So how do we deal with that?
So right. So we had there's then I want to, with respect to the work we did in DESeq2, I want to make sure to acknowledge like the other work that had been done. So first of all, there'd already been the proposal from microarray analysis packages to share information. That was clearly a good idea. That you look at tens of thousands of variance estimators for limma, look across tens of thousands of variance estimates, and you get a good sense for how spread out they are and what the central value is. And by moving them towards the central value, you get a more stabilized test, and you can show that it's more powerful. So there will always be noisy variance estimates in the tails. It's actually related to another concept in statistics called winner's curse, that if there's a lot of noise, then if out of a distribution, the top is probably not the top. It's probably just got lucky. And so there will be noise in the tails. So we move things towards the center, and we get more robust and more powerful testing. So limma already exists for continuous value. And also there had already been work from the EDGAR group, so Mark Robinson and others, to move together dispersion estimates in the context of this gamma Poisson or negative binomial.
Right. And even a simpler solution would be just assume the same dispersion across all genes. What was the problem with that?
Right. And so the first version of DESeq had kind of a balanced approach where they a balanced and conservative approach where they look at the distribution of all the estimates of dispersion across all the genes, and if they are high, if they're on the top half of the distribution, just leave them be, because that's a conservative choice. Because if the dispersion is high, we make it low, we might be tricked by noise and think that there's a significant difference when really there's just a lot of variability within groups. But for the dispersion estimates that were on the bottom half of the distribution, they moved it up to a common value, which is the middle. So they moved it all the way up to the middle. So it was kind of information sharing for the bottom half, and then conservative choice for the top half.
Was the top half also consulted to infer the middle?
Yes. Yeah. So look across all the genes. Although there was I'm trying to this now. You're asking me to think back to the algorithm details in 2012. So I believe at that point, Simon had implemented a robust procedure for defining which genes inform the middle. So there was kind of iterative process of defining the middle, and that's actually pretty important. And that's actually still an interesting topic. I've seen really interesting papers like from the single cell world in, I think, the basics. One of the basics papers looked at like how should outliers inform the prior. That's a really interesting topic. And there's also really interesting work from the limma group on that. Belinda Phipson is, a lot of people have thought about that, and that's a really interesting area for statistical research. So Yes. So your question was how should the top half? It does inform the middle, but then maybe not all of the points. So if some of the points seem very extreme, then maybe those should not change your concept of like where's the middle of this distribution.
And so, what's the problem with just pinning all the dispersions to the same value, be it the lower half or just all of them?
Yeah, here the problem is biology. It's that um, genes do have different variability or dispersion. So they just some genes have more for whatever reason like tightly controlled variability across mice. Let's say we keep going back to these mice. So some genes are kind of more tightly controlled, maybe, and others we just do see more variability across. And so, plugging it all to the same value, we would also be making mistakes. So I said the MLE, our first, our just looking at the gene estimate is noisy, but we would also be biased if we just plugged in the middle value. And DESeq, the first version, it had made a choice. It said, okay, we'll accept bias for the low estimates with respect to the whole distribution. We'll be biased on the low part. We'll be unbiased on the top part, but we'll be conservative because we don't want to move them down. Moving them down would produce lower p-values and maybe some false positives. So we accept false negatives by moving the low estimates up, and we try to not induce false positives by keeping the high estimates high. But that is conservative. That procedure, they knew that, and so at that time they were interested in kind of updating that to be more balanced, so more of a shrinkage approach.
Yeah. So what I assume has happened was that these people started to get a lot of angry letters like my p-values are too high, like fix your software.
I can't speak to that, but that's a really good point. There is this challenge in writing methods to take input. Users give invaluable input, and they can spot problems, bugs, method gaps. It's a challenge to be receptive, but then to also hold fast to we've come up with this set of methods, and we have some theoretical justification, and if we let things slip, we will lose our grasp of what's going on. That's a really hard challenge.
Yeah, especially for a package as popular as DESeq and DESeq2 have become.
Yeah, and then the other as time goes on, and one of the most important things I've tried to do is not change things too much, because then people expect it to be like a fixed quantity, and they've they accept they expect a certain performance, and so in the early years after it published, like a year after it published or two, there were things that I wanted to update, and we made updates, and there was a lot of angry emails about I updated my R, and now I get a slightly different number of, in my opinion, slightly different number of adjusted p-values, less than a threshold. And in my opinion, it's like, oh yeah, but it's kind of similar, right? It's like went from 15 to 18. That's not, but for an investigator, with those three might be really important, and they want to know, are they?
Yeah, those are yes or no decisions.
Yeah, those are 3 experiments to perform, and they want to know, like, well, which should I trust? And as a method developer, you develop methods that have certain operating characteristics. So you're like, well, like overall, it should have these expectations, but I can't tell you if that gene is to be trusted, singled out.
That really shows you, how differently various people interpret these p-values, right? You, as a statistician, you have a more theoretical understanding of a p-value, which is arguably, it's definitely more correct, right? But there is this folk understanding of p-values and tests that give you this hard yes or no answers. And I don't know, do you have any thoughts about how could the field, has the field evolved past that?
Well, I think that the concept of the false discovery rate helps a lot, because then it's a property of a set. So we're no longer looking at hopefully people are no longer looking at an individual p-value and saying like, do I believe that one? But it's getting people to think about sets and rates. So then they understand that, we're not, the method's not going to say these are the, if we say 5% false discovery rate, these are the 5 out of 100 that are false. Maybe it's easier for people to get the idea that like they're in there. There's some probability. We expect on average there will be 5 out of 100, but I can't tell you which ones. And I also see that. I think it's really good also to see that in GWAS. People talk about like posterior inclusion probabilities and credible sets of variance, because we just can't say which one, but we can give you a posterior probability for this set.
Mm-hmm. I don't know. I still think you're a bit optimistic on that front, and I talk to a lot of people who treat the multiple testing correction. They just treat, those were incorrect p-values, but these p-values are correct, and so if these p-values are less than 0.05, then this must be true, or something like that.
Yeah, I guess like I try not to blame, when there's this lack in an understanding, I try to think about it as a communication problem, and just that we need to do a better job with outreach and teaching, and maybe we need more tutorials that are directed at biologists to think about what are these quantities you're getting out of these software.
Which leads me to a question basically about the Bayesian approach. And would you agree that Bayesian approach didn't really achieve any significant adoption in this field? Meaning that there are some approaches inspired by Bayesian inference, like what you basically mentioned, right, about shrinkage and priors and maybe some empirical base, but that's still not a full Bayesian approach that relies on informative priors, that relies on analyzing the full posterior distribution instead of doing statistical tests. So, and it would seem that the Bayesian approach is exactly what this field needs because of the small sample numbers. So why was that not successful?
Ooh, I'm gonna get in trouble no matter which way I go with this one. So, many times I've written methods that draw heavily on the Bayesian framework, and I even call them Bayesian methods. But then someone else might say, but they're not because of what you just said. Actually, like you don't prior is not specified ahead of time, and then I make approximations here. So I think a key aspect is that the data generating mechanism, and this is something I really like about Bayes, is that you think about it, you write down the data generating mechanism as part of coming up with a model. It's very complicated, and there's many layers and dimensions. And think these single cell Bayesian methods, it's these people, I think I really like all the Bayesian methods in single cell, and you look and it's incredibly complicated, because you have samples, cell types, cell states, trajectories, pseudotime, cells, genes, etc. So there's just lots of layers. And you want to be flexible in how you specify the data generating mechanism. So you're going to end up in a territory where you have to do computational sampling from the posterior. And so that is the right thing to do, but it may give you some desirable properties, but it leads you to a place where your software just requires inspection of diagnostics and things like that. So it limits its applicability to so who can run this. There are certain things that you need to check. There's certain things you have to ensure convergence and that you've sampled enough. There are lots of important things to check and to know how to interpret the output. And so, and then so then it limits who's qualified to interpret and understand and read the output of this method. And then another aspect is people want deterministic output. So that's another thing, is that you could have different runs will give you slightly different answers. You could try to avoid that by running it longer, but nevertheless, it's not deterministic anymore. And that's another thing that people really want, that I run it and you run it, and we and there's a kind of deterministic state. So you can set seeds, but just the idea that it would be dependent upon a number at the top of your script is undesirable to some people.
But you think overall the reasons are more technical rather than fundamentally people don't agree that Bayesian methods would be better in this case. So like if we were in a different state of technology, if inference was fast enough that we could have a huge sample size and like efficiently analyze it. So effectively your results would be mostly like deterministic. If those technical issues just suddenly got out of our way, would Bayesian approaches, do you think, become more attractive in the science?
Well, so that's another thing, is like as the sample size grows, then most of these Bayesian models then convert, like the prior goes down and you end up with the likelihood dominating. So then
No, sorry. What I meant to say was like the posterior sample.
Oh,
right. So how many samples you can reasonably draw from the posterior?
Oh, I see. Yeah, I think it's a good question, and I think one thing is, I think there have been people who've done really good work to make it more broadly usable, like the Stan team, and I've used Stan many times in my research, which is a software suite for sampling from posteriors from models that you write down, and they and previous groups like Bugs and Jags have done a ton of work to make it more accessible, and also to teach lots of people, lots of scientists in different fields, how to do the diagnostic work that you have to do to interpret the output. So then I think there's like a gradation of this, how well has Bayes been taken up in computational biology. There's lots of methods that are doing this kind of approximation to empirical Bayes technically, where we look at the data a certain way to over many, many features to form a prior distribution. And then like DESeq2 is then calculating p-values after having, improved estimates with using Bayes ideas, or even doing Bayesian computation, but using the data to inform priors. I think that, those are really popular. And then I think there are people who are directly, running Bayesian models from start to finish. It's just that those are hard to distribute as software.
Speaking about Bayes, here's what it would seem to me a simple thing to do. So you mentioned that we cannot assume the same dispersion across genes because, the biology, because like different genes are apparently have just different dispersions. Why is there no simple way to aggregate the information, not just in so if I understand correctly, all these packages, they only pull the data within the data set, right? Whereas we have by now a ton of already performed experiments. Why not pull data across all of that data and effectively form a very good, very informative prior?
That's a great question. And right, it's not done that much. With a caveat, I think it is done in deep learning, that people start with a good map, start with a good network architecture learned on another data set. So transfer learning that idea is used a lot, but yeah. So why don't Bayesian models pull from the SRA? I guess one easy answer is the amount of experiment to experiment technical variation. So if you had So batch effects, the fact that like the shape and the location and the scale of the data would change across different experiments. So just because I have, control samples of this organ in this organism, I can't immediately assume that the distribution of those same samples in some other lab would be a good fit. But there must be some methods that do that. Although I don't use them I don't think I use them in my practice. But
Right. I would think that there wouldn't be a problem at all because by definition, like you form your prior over many, many experiments, and your experiment, your particular conditions that exist in your lab is just a sample from that distribution. So by definition, like if you analyzed enough experiments, you would get a proper sample from that distribution.
yeah, that's a good point. Yeah, I mean, I'm thinking, so I was a postdoc in Rafa Irizarry's group, and he and Matthew McCall and others had a paper where they looked at the question of, is a gene expressed? And you have a sample and you ask, is the gene expressed? And they did pull from GEO, so gene expression omnibus. They say, well, here's this gene's expression in thousands of samples, and we have a distribution, and we'll just like look at it relative to that reference distribution. And that's a really powerful idea. So and there are some methods that do make use of reference samples. I mean, just to plug a paper from my group recently, we were looking at splicing. Like we have a sample, but we maybe have low counts or not many samples, and we want to compare its splicing pattern to a reference panel because we don't have many samples. So we use GTEx for this because there's thousands of samples in GTEx, and they have provided isoform level quantification. So different isoforms of a gene, and then they estimate the relative proportion of each. And so we tried to use that as like a reference distribution to say, what is the splicing pattern like? Is it similar to heart or to liver? And like I think our method would be better if we used an even broader panel than GTEx. I think because we have these short reads where there's ambiguity. There are not, as far as I know, like there's so many different ways to try to estimate that, and there's variable uncertainty. So it's harder than it was with GEO to just have like a gene expression table. You just had the probe value, the probe intensity with microarray. But so that would be cool, though. It would be nice if we'd looked across like all the SRA samples to characterize the splicing in a sample.
Okay, so we derailed a bit, but so you described how DESeq, just DESeq1 version 1, it did this half pulling where it pulled the dispersion up from the lower half but left the upper half intact. So how was DESeq2 different in this regard, and what was the push behind changing that behavior?
Sure. So Simon and Wolfgang, like they knew that the choices they'd made were conservative, and then they wanted to like have higher power, and also ideally just more accurate dispersion estimates, because they know you've introduced bias if you move all the lower half up. So then the idea was to have a method which would just again like apply a Bayesian framework. So estimate a prior distribution for the dispersions for these individual gene dispersions. We don't know them. The Oracle didn't give them to us, so we don't know them. But we could try at least to estimate their distribution, and then we have the likelihood for each gene, and so then we could get a per gene posterior estimate. And just to mention the existing methods, this was like summer of 2012 that we were in contact talking about like how to update this, what would be a potential project to work on. There was a method called DSS that came out right around then, which basically did what we wanted to do. It it applied a Bayes formula to get a posterior estimate for each gene. There was also EdgeR, which had a weighted likelihood. So you had the per gene likelihood and then a weight applied to the common dispersion value. And the way at that time, the way that they balanced the per gene information and the across all genes information was with a parameter that was actually, I think at that point it was fixed. So it was like the weight across the common weight for the common dispersion estimate was like 10 times, like looking at 10 genes. And so we were kind of in this space between these two methods. We wanted to have it be the weighting of the prior should be determined by the Bayes formula. So we didn't want to fix that value. And then the space where we were trying to fill with DSS was that we thought we could estimate the prior by looking at the data. And DSS had done this, I think, with simulated data. So at some point, they had a step where they simulated dispersion values to estimate the width of the prior. And Simon had the point that we could estimate it from the data using some distributional assumptions about these dispersion values.
So basically, first you look at each gene separately and calculate the dispersion, what it would be if you had just that information, and then you try to fit some kind of parametric distribution to these values.
Yeah. And so I think the key idea for us was that on the log scale, so DSS had also looked at the log of the dispersion values and said, well, that looks like a bell shape for many experiments. So we could try to say that it's normally distributed on the log scale, these dispersion values from each gene. And then I think Simon's point when we started this project was that on the log scale, we could figure out the prior width because it should have a certain distribution. So the log of the dispersion values should be chi-squared with the degrees of freedom that had to do with the degrees of freedom. So the sample size and the number of groups or the number of parameters in the model, that would tell us what the variability of that distribution should be if they all had the same dispersion. But then when we see more than that distribution, that tells us about how variable they actually are. So if all the genes had fixed dispersion, we had a formula for how wide our observed log dispersion values were. We see it's wider than that, and that difference is what we use to estimate the prior width. And so one more detail, I know there's a lot here, but the fact that if you start with a distribution that has a certain width and then you have extra sampling variability, that's just a property of normal distributions. So if you sample a value and then you from a normal distribution and then you sample again, this hierarchical two times, the variances just add. So that's a really simple formula for if you see something this wide and you think it should have been this less wide, I shouldn't be using my hands in a podcast. It's less wide, then the difference is your estimate of the width of the original prior.
Interesting. But how do you validate a method like that? Because many people may have many different ideas how they want to calculate these dispersions, but ultimately you make a claim that your p-values give a certain false discovery rate. How do you ensure that that is a valid claim?
Yeah, and that's a great question. Generally, how to validate methods, statistical methods, or so the first iteration of this paper. So we implemented something like I just described, and then the next question was how do we validate this? So the first round, we thought to do data splitting, and that was tricky because there were still not so many data sets that had a lot of replicates. So Now there are more data sets that have like many replicates. So there's a paper from Sturch that has like 40 replicates of yeast per in two conditions. So that's really nice for benchmarking in any context. Like if you can find high replicate data sets, those are really nice because then you can split the data and you can look at the set that you say has a certain difference, and you can estimate like the difference, like the fold change, say, and then you can compare it to data that you haven't looked at. And that's just my favorite. That's my favorite approach. We didn't have too many samples, so we tried that. But then if you don't have too many samples, just because it's not called in the held out data doesn't mean it's false. It could be that in the held out data, it was a false negative. So that's problematic when you have reduced samples. But we did try that.
But still, how would that method work? Because you have to make the judgment call what is different, what is not, right? So it's not just a matter of like under a threshold, over a threshold, right? And it also depends on your sample size. Presumably, if you have a high enough sample size, everything is statistically significant. So it's more like a philosophical question, right? What does it mean to be differentially expressed?
Absolutely. Yeah, absolutely. I think it is really hard in this point null. When you're talking about a point null, which we just mean like the null hypothesis is that the difference is zero, or that the fold change is one. That is hard, and that is one reason that I like, and we've tried to move towards this evaluation criteria proposed by Matthew Stevens called the false sign rate. That's then, if you have a posterior, it's the integral of the posterior distribution on the other side of the sign. So if you estimate there's a positive sign that the gene makes the gene's expression goes up, what's the probability you got the sign wrong? It actually goes down, and that is really well accommodated to the data splitting, because then you can just look at some other data and calculate the sign, and then the rate of how many times you got the sign wrong. Whereas, so you're not, like you said, this like point null, is it ever true? Maybe the point null is true for no genes if in an experiment, but you certainly will get wrong signs.
Right, and so you said that this approach wasn't really applicable because you didn't have enough replicates in the existing experiment. So which one did you take instead?
Yeah, so we did try the data splitting, but with a bunch of caveats. So we tried data splitting, and I forget how we determined like if it was if it was replicated. One thing, and I think there's a paper from like Roger Ping and some people at Hopkins about like you should probably be a little lenient with the p-value in the replication data set, right? Because it's not necessarily going to get a 0.05 p-value in the replication. It doesn't mean you didn't replicate it. But anyway, so I think you should think about, even if you're trying to do the data splitting approach with not so much data, you should think about how you define the outgroup. What we did, so we submitted it with like a highly caveated data splitting for the evaluation. And then the reviewers said, no, you have to write a simulation. You can't publish a bioinformatics method without a simulation. And we had already done simulations in working on the method. So that was actually pretty easy to add in, because we had, as when you're developing a method, simulation is incredibly valuable. Like it should at least do as well on the model that you think the data follows. So we had already done a lot of those, so we could pretty easily plug those in as a simulation as well. And then then we put, I think in the order in the paper, simulation then comes first, and then the data splitting example.
So were there any other significant changes between DESeq and DESeq2 apart from the way you sort of pulled the dispersion estimates?
So that was kind of the biggest. That was like the first order of business. And then aside from that, there was another methods development, which was to basically do the same procedure to the fold changes. So there were some like fully Bayesian models, like BaySeq and ShrinkSeq. There were other models that had moderated or applied Bayesian shrinkage to the fold changes, the effect sizes. But I think they both relied on sampling. And so we provided another kind of non-sampling based approach to get Bayesian estimates for the fold change. And so and why bother with that? If you have p-values, you can go and, you can just look at the significant genes. And our argument there was that I think the Bayesian effect sizes are good for ranking. So if you want to rank genes not by their FDR set, so like their true or false status, but just by their fold change, we've argued that the Bayesian estimates are good for ranking genes by effect size. So that was a methodological difference. And then the last thing was we wanted to switch the data container to this new thing at the time called a summarized experiment. And so Bioconductor had introduced this new container from the expression container, the microarray expression container. So they were moving to a container that had the possibility to have genomic ranges tied to the rows, and I think that's really cool. I always thought that was a big improvement, because then you can do range-based querying of the object. So you can say you have this big object which has lots of genes, and you can say, show me the genes that are around this particular region, like say this GWAS hit or this ChIP-seq peak, and you can just pull out based on that range the genes nearby.
And after DESeq2 was originally released, you mentioned that you try not to do any big changes, but have there been changes that you've made or wanted to make to DESeq2?
Yeah, so there were. I did make changes to the dispersion in the early period, and I tried to document these, but like the first few releases, there was just a lot of complaints about it results changing too much across version. And so in my opinion, the last big change was to, at one point, this was kind of like as I was in my postdoc and planning to start off as a PI, I realized that, well, one thing I want to do is work more on the effect size estimator. So in the first version of DESeq, we had used a normal distribution, and a lot of people, this is again, this example of good feedback. We had gotten a lot of examples of like that not being a good distribution. So like people had experiments where one gene was targeted and should be very highly differentially expressed, while the rest should not be affected. And that didn't really look like the data sets that we had designed the software for, because so then that's not a normal distribution that we were assuming the effect sizes could be approximated by normal, and that's not at all normal. So one of the first things I wanted to do was to like explore alternative, more heavy-tailed distributions. So I did make a change at one point to split the effect size moderation out of the main procedure. So the main DESeq function would just do the dispersion shrinkage, but not do the effect size shrinkage. And it would give you, p-values and adjusted p-values. And then the second procedure to shrink the log fold changes would be a dedicated function. And that then allowed me to provide a switch where you could look at different methods. And we also, another convenient, nice thing about that, splitting it out to a separate function, was that I could also put in like, there's an adaptive shrinkage method from Matthew Stevens called ASH that we could plug in that as well. So you can get this kind or that kind or that kind, and other ones potentially by making it like a separate function.
And so these days, if someone does a differential gene expression experiment, so I guess the main alternatives are DESeq2, EdgeR, CuffDiff from Cufflinks. What else?
Limma-Voom. Yeah, so I think Limma-Voom is really a strong method. It's also very fast because it has under the hood the linear model. So yeah, I think there's these are all really good pipelines.
What would your advice be if someone had to choose between these? As biased as you are.
Yeah, sure. I'm definitely biased. What I actually tell people when I work with them is, if you have a pipeline that's working, then you understand the software and you can interpret the output, then you should stick with that. Among methods that have been, looked over quite a bit. So like, the methods that have been benchmarked and we understand this term, the operating characteristics of those methods, then and you've got a pipeline that works, is to stick with that.
Yeah, but if someone starts from scratch.
Yeah, so if they start from scratch, there are some subtle method differences. So like if they're working with me, and I would argue that it's useful to like at some point look at the effect sizes and rank things by effect sizes. So I'll often, and I also like to look at those plots. So the plots of the posterior fold changes. So, I'll put them down this path. Also, the other thing is like, when I collaborate with people, I have the ability to add things to DESeq2. And so like, that's another reason why, in collaborations with me, I often will use DESeq2, because I can like add in something every now and then. There's something I notice that can be modified, but I try not, it's always like, I try not to change the default now, but it's like, a side path.
Are there any circumstances where you would say, DESeq2 is not the right choice for you, instead use that one?
Oh, sure. So I'll give you some that these are documented cases because I say these all the time on the bioconductor support site. Here's one is people come with an experimental design where they want to compare across two groups. I'll make it as simple as possible, but there are within these two groups, they have multiple measurements on a unit, like say a donor, human donor. So you have multiple, not technical replicates, but biological replicates for each human donor. And then they want to compare across the two groups. So they want to take into account the structure within group.
So it's like a hierarchy.
Yeah. And so you would, if you were approaching this outside of bioinformatics, you would use a mixed effects model, and you can, account for the fact that the samples from the same donor are going to be correlated with each other. And so in DESeq2, and I think also in EdgeR, there's not a way to account for that. We only have like fixed effects GLM. And in limma, there is a method to account for that. So that's really convenient. And so from my understanding, that the statistical methodology had been developed for multiple probes. So the fact that you might measure, so in the different dimension of the matrix, you might measure the same probe might be correlated within a gene. That method is called duplicate correlation, but it also extends, you can apply the same math to the columns of the matrix. So you can say these samples have a correlation structure, and the method learns the correlation from the data and then uses that for the inference. So that's one. And then a much easier one to describe is that when you have a lot of samples, then the GLM will be slow. And if you're not really interested in the very low counts, so I think this doesn't work for single cell because 0, 1s, and 2s are not to be thrown away. But if you're not interested, if you're looking at bulk RNA-Seq and you have very many samples, limma is going to be really fast. So I myself use limma-voom when I've got many, many samples, and I recommend the same thing on the support site to people.
You must get a ton of like user support requests. How do you deal with them?
Yeah. So I get maybe like, I think I calculated it the past few years, and it's like 2 to 3 a day through the support site. And so, one thing I should say is that the fact that Bioconductor at some point transitioned from a mailing list to a support site, and that's been really huge as a developer these don't go through my email inbox, or like they do, but then they get moved to a folder. That's just really helpful. And I can go and edit my response. You can block code and have threaded discussions, etc. You can unsubscribe.
So support site is something like Stack Overflow, but specifically for your package.
Yeah, and it's actually, it's for all Bioconductor, and it's based off of BioStars. So Istvan helped a lot in getting this up and running for Bioconductor. It's the same source code. It's like a forked version of the source code, I guess. And that's been really great. And Yeah, so Lori and Martin, and there's a couple of people who helped out, who made this possible from Bioconductor side, and Isfan from BioStars side. That's been really key. But then, just to say how, from the beginning, I tried to answer everybody and try to make everybody happy. People came with questions which were really not about the software, but more about like, how should I design my experiment? And I, in the beginning phases, I just wanted people to use the tool, so I would try to answer everything. Now that's really not possible because I have PhD students and teaching obligations and a family, and so I have to sleep. I've tried now to transition from answering literally everybody's question to, I just try to zero in on the software based questions. And when it's a question it's a valid question, but it's about, how should I conduct my experiment? What would be the right way to think about these samples? I have to push back on those and just say, I don't have time. It's basically statistical consulting. That's another of my job actually at UNC, is I'm a statistical consultant, and I have, that's a part of my time. Is I have to, back in before pandemic, like, people come to my office and we chat about statistical design, but that's like, I'm actually, that's part of my supported effort. So I have to push back on it and say, maybe look around at your institute for consulting cores that you could sit down and talk to someone.
Does anyone accept you do any user support for DESeq2? Do you assign your PhD students to do that or something?
I haven't asked any students to help. There are a number of people who help out, who are a great help. So there's Alexander and Kevin, and there's a couple of people who really help out quite a bit. They field various questions about RNA-Seq in general, and then some about DESeq2. I haven't asked any students. I mean, the way I think about it is like, if you get into a PhD program or whatever level, and you become interested in developing software, that's great, and you will benefit from providing support for your software. So, I have this talk with different trainees that I work with, or I'm on their committee, that like, it's a lot of extra work. But if you want to stay in this zone of tool development or software development, then it can carry you pretty far if you build a community of users. And one way to get that community is to be responsive. But it depends. If somebody's developing methods as part of their dissertation, but then they want to go into a different field, or they want to transition, then dedicating their lives to answering user questions might not be the right answer for them.
DESeq2 is not a new tool, but part of the reason, also that it's been a very influential tool, but the reason I wanted to talk to you about it is that you recently received a grant from CZI, and maybe you can explain what that is and what the grant is for, and what does it all mean for the community?
Sure. That's a good point. So Chan Zuckerberg Initiative is CZI, has had a lot of funding opportunities for development of software, bioinformatics software. Some of them are specifically tied to single cell or imaging. So like developing open source tools for single cell analysis or for imaging. But then there was a general call, I guess it was more than a year ago, just for open source software that is essential to scientific workflows. So that was the EOSS, Essential Open Source Software, RFA, and they had 3 different rounds, and it's a really unique funding opportunity. There's not often funding available for existing software to maintain it, or to answer user emails, or it was very flexible, the kinds of things you could use it for. And so I applied for to extend DESeq2 in 2 different ways, and was lucky enough to be one of the awardees. And the two, I'll just briefly describe these 2 proposals. One was to and this is work that's been done by Kwame Forbes, who's a post-bac scholar at UNC. So he and I have been working to create a function to help people with bulk RNA-seq expression data connect their data to single cell data sets that are on Bioconductor. So a number of people have processed single cell data sets and then made them available within Bioconductor. So it's easy to kind of download those and get the single cell counts in your R session. And so we just wrote, I mean, it's not theoretically complicated, but just some bridging tools to help those people be aware of those data sets. And then they can make a choice based on like, okay, you have some human data, so here's some human single cell RNA-seq data sets, and they can then pull them down and then like look at, for example, a differentially expressed gene in your bulk data, like what cell types is that expressed in some other single cell data set. So that was one of these proposals from this CZI. And then the other one is something we're still in the early stages of development. It's software for generating this like null set of genomic features. So I guess if people are familiar with bed tools, there's a tool within bed tools called Shuffle, which will move the features around in the genome so that you can compute. If you're computing like an overlap statistic, you might want to know, well, what if we just shuffled the features around? How many overlaps would we have then? And that you can then, if you do that a bunch of times, you might get a null distribution of the number of overlaps. So within Bioconductor, there are also packages for doing this, for permuting features, coming up with like a permuted distribution of features. And we're just trying to make a little more options on how those features can be distributed. So like nearly everyone allows there to be an excluded list. Like you don't want to put features on the centromere. You don't want to put them, there's certain places you don't want to place features. But then additionally, we're looking at two aspects. One is that features tend to be clustered next to each other. So there's some statistical theory from Peter Bickel and others from Berkeley on sampling blocks of data. So that's called a block bootstrap. If you grab blocks of data, and it's been used in time series really effectively to generate like artificial time series, is to grab blocks of a time series. So they showed that you could do that for genomic inference, and that's really has some nice properties. And then another aspect that we're looking into for generating these null feature sets is controlling for covariates. So you might want to go look at some background distribution of features, but matching with respect to a certain number of covariates, like GC content or gene density or something like that. So it's not the second idea. It's not a whole package that does everything, but it hopefully will just be like a modular package that sits within a bigger framework using other packages that helps you do enrichment analysis.
I can't help but notice that although you said there's not enough funding to support existing software, what you've been talking about is like writing new code, new software.
Yeah. So it's not maintenance. You still have to get the award. So you still have to like come up with some new ideas. Well, they were interested in helping people who have created successful software to extend it. And I have had troubles, sometimes publishing papers. You extend something and then it could be rejected because it's not sufficiently novel. So like, I think that aspect is really helpful to give you a little cover to say, well, we're extending this work, but we're not saying it's brand new. And it's going to enhance the features that already exist in this software. I just mentioned another great thing about this program is that they're now focused. They have another RFA for increasing diversity and inclusion in bioinformatics, which I think is so important. And so getting more representation of people from, let's say, underrepresented backgrounds is kind of a catch-all term. But basically, we all need to kind of do our best to help increase that. And they now have some new RFA for proposals that will try to increase diversity.
Cool. And to wrap this up, do you have anything else you wanted to talk about, or maybe talk about what you are up to these days?
Sure. Yeah. So just briefly, I think I've tried to diversify lab interests, and when I got to UNC, there were the genetics department where I'm co-listed in the genetics department, and they have really strong a lot of investigators with QTL data sets. So one of my, outside of RNA-Seq, one of my areas where I've been trying to do more methods development is in EQTL and the integration of EQTL with GWAS. So I guess, yeah, my latest thing that I've been devoting a lot of time is a Mendelian randomization method called MR locus, which helps to within a locus try to estimate the effect that a gene has on a downstream GWAS trait. So, but there's also again like there's so many methods. There's a lot of really great methods that are coming online right now to do that same task. So, you know, there's a lot of other great tools to look at there.
Well, Mike, you've been very generous with your time, and it's great to have you on. Thank you for being on the podcast.
Thanks so much, Roman. This has been a great pleasure.